Hello, Mr. Novak here again. Today I'm going to talk to you about mutations, okay? A mutation is when a cell makes a mistake when replicating or transcribing, okay? That mistake can be a number of things, and that's what we're going to get into here today, okay? Generally speaking, there are repair mechanisms to help fix these mistakes, and some other proteins will come through and some enzymes will come through and kind of double check, like spell check, just double check the strain of DNA. There are times when that change is missed, right? That mistake is mixed and it's not fixed. And that leads to a permanent change in the DNA, which is what we call a mutation, okay, when that change stays there forever and ever. And then recall, because replication occurs, that change is going to then be in every single DNA strand from there on, okay, when that DNA replicates. A couple of things about mutations is that they can be spontaneous, right? DNA polymerase might just add the wrong base pair. It happens, okay? There are other things, chemicals, radiation, and whatnot, that can also damage DNA, okay? Whether you ingest it or hang out in the sun too much or whatever it might be, those chemicals and radiation can also damage your DNA, which then can lead to mutation. Okay, so here you can see there are tons of different things that can cause mutations, right? If you ever gotten an X-ray done, they put that lead vest on you, right? Because that can help damage your DNA, just like the sunlight. The chemicals in cigarette smoke or vape and all that can do it as well. You've heard of like carcinogens, right? Things that cause cancer. Those are all mutagens, right? So a mutagen is anything that causes a mutation in our DNA. So you can kind of see here how like radiation might work. So if you're hanging out in the sun a lot, radiation, the sun's UV, right, will come in and it'll break those bonds. Those hydrogen bonds are really weak, so it'll break them. Okay, once it breaks them, those base pairs are free to kind of do a couple of things. They can pair with bases next to them. They can just damage themselves, and so a lot of things can happen, right? So you see in this picture, right? Picture one, I'll call it, right? Here's a normal strand of DNA, normal protein gets made. Um, if we get that mutated gene or that mutated base pair, what happens is that you can create an abnormal looking protein or just no protein at all. We'll talk about kind of how that goes in a second. Mutations can occur in a lot of different spots in your body. We have these cells, they're called somatic cells. Those are your body cells, like your skin cells. Okay. If you get a mutation in one of those, each then new DNA strand, each mutation will then get passed on to each dotted cell. It doesn't necessarily get passed on to the offspring if you're going to have children, but it can then get just in each individual cell. If these changes, these mutations happen in your gametes or your sex cells, those eggs and the sperm, they will be there and you will pass those on to the offspring, to children, and then they get it in every single cell. So kind of depending on where these mutations occur, you can have big effects or not such big effects. So let's talk about different types of mutations. Okay, the first type of mutation we have is a point mutation. A point is like just one base pair changes. Okay, this could be a substitution where you just replace one base pair with another. If there's supposed to be an A, maybe they put down a G instead. Okay, we know from our amino acid chart that there are a lot of amino acids, or there are 20 amino acids, and some of those codons code for the same amino acid. So if it codes for the same amino acid, that change happens and it codes for the same amino acid. It's called a silent mutation. We don't notice it. Okay, you'll still make the same protein, and you'll be good to go. It could be a missense point mutation, missense substitution, where you get a totally different amino acid. All right. If you're thinking like, oh, I need this amino acid, and you get a different one, it could change the whole structure of the protein. It could make the protein non-functional. It could be a nonsense mutation where that change in the base pair creates a stop codon. That stop codon then terminates the protein being made early, and you're not even going to create a protein there. Okay, you can create something that's just going to be non-functional, and your body's probably going to destroy it. Okay, the other type of point mutation we have is called a frame shift. Okay, a frame shift is when you get an insertion or even a deletion. Sometimes they don't put down a base pair, so you lose it, or you duplicate a giant section altogether. Okay, when that happens, the messenger RNA is not read correctly at all. You get these non-functional weird proteins, and bad stuff happens. So kind of looking at some pictures here. Here's a substitution, silent, no effect, right? So this top one up here, right? This is our original strand. We see it makes these 4 different amino acids, and we get a normal protein, okay? Down here then, right here, we see that that A, right, is put in it instead of the G. Okay, what does that mean? That means that A, when it gets transcribed, becomes a U. Okay, when it gets translated, right, we still have these same 4 amino acids. So that'd be a silent mutation. Nothing happens there. Okay, we still get the normal protein being made. Here's a missense mutation, where again, now instead of, right, we get that T inserted in there, right? Instead of the C, what does that mean? That means that this becomes serine versus glycine, and we get a non-functional or just maybe a different protein altogether. Here's a stop. Here's a nonsense, right? So this A, right, got inserted instead of the T. That created a stop codon. So we don't get these 3 amino acids being made. It stops the protein, doesn't make a protein. Here's a frame shift mutation. Okay, so here's our normal, right, strand up top, right, normal strand, right, that's what we start with, ACG, AGG, so on and so forth. Here we inserted, right, and we get something totally different. We insert a amino or a base pair, and it shifts everything. That's why it's called a frame shift, okay, because we've shifted, and now we're off a base. Okay, now we have an extra base, just some random extra base. Maybe we don't get that stop codon that's supposed to be made, and this protein will just keep continually going. Or maybe it actually shifts, and now you have a stop protein or stop codon somewhere random. Okay, here's a -1 kind of frame shift mutation down here, right? And you can see that again, we get these extra bases, and so we're not making the correct order of amino acids, which means we're not making the correct protein. Here it is again, just another picture of it. Here's causing extensive, it says extensive missense because again, we got these first ones are the same, and then these other ones are just off. Now everything from there on is going to be off and wrong, and you're not making the correct proteins. Here's another kind of one for that. Some examples of this. So sickle cell is due to a point mutation. So you have first sickle cell anemia where the blood shape is different. It can't carry oxygen and stuff properly as it should. Okay, so it is a point mutation, right? So one amino acid, excuse me, one base pair is different, which changes one amino acid, which means we get a totally different shape in our blood. Okay, and so we can see that this is caused by a genetic defect. It's not carried by a lot of folks, but it's carried like 25% in some regions of Africa. So again, we see this in certain populations, right, where this mutation occurs and then their offspring have it, and it gets passed on kind of generation. Okay. So some other pictures. Here's a duplication mutation where you see we took this huge chunk of DNA and replicated it here and accidentally replicated it again. Now we get this extra large chromosome. Okay. Just a quick example. Here's our DNA sequence, right? So I'm going to break this down. I got 1, 2, 3, 1, 2, 3. So we have 3 codons, right? The messenger RNA would look like that, right? 1, 2, 3, 1, 2, 3. And our amino acid sequence would look like that. Now, a mutation occurs, right? So I go 1, 3, 1, 2, 3. If I go back, that first one's the A's, right? This was an A, and now it became a G. So we get our amino acid sequence, or excuse me, we get our messenger RNA strand. It looks like that. I'll do that, and our amino acids, right? And so the amino acid sequence, if you go back real quick, leucine, histine, and stop, it is now leucine, histine, and stop, right? This is called a neutral mutation. Okay, so nothing happens here, right? We talked about that a little bit. I just want to kind of show you how that would work again. So nothing happens, and this happens quite frequently. You would never know you have a mutation if it did this. Okay, and that's what I have for you for mutations.